This presentation is dedicated to the Hungarian Academy of Sciences 200th anniversary. Discovery and development of new drugs is a very complex process. It involves many scientific, engineering, and medical fields. It always starts with a lead identification, which in the recent years, due to genomics and combinatorial chemistry, a dramatic increase in the number of possible targets. However, no corresponding revolution took place to lead optimization and the development process. We are proposing that retrometabolic drug design represents an important solution to reduce the time and expense needed for drug development. Let me point out that a drug undergoes always metabolic transformations in the body, which results multiple metabolites, active metabolites, analog structural metabolites, reactive intermediates, and inactive metabolites. We propose that desirable metabolic pathway should be designed into the structure together with the design of the activity. So the focus on not on the improving the efficacy but increase of therapeutic index, which is a ratio of the toxic and effective dose. This resulted in the so-called retrometabolic drug design loop, in which a drug can be designed to be part of a chemical targeting system, CDS, which is a biologically inert molecule which is converted to the active drug by several important, several enzymatic or chemical transformation. It is not equivalent to a pro-drug concept. This enhances the drug delivery to a particular organ or site, and this is accomplished by attaching the drug molecule to two types of functions. One is the target, or that is the most important, and the rest are the modifiers. One example I give, and we have designed many different organ-targeted systems, designing drugs which are targeted specifically to the brain. Here we use the difference between the capillary walls in the general circulatory system, which the capillary walls have fenestra, allow different mechanistic procedures to allow the circulating molecules to get in the general the other components, as opposed to the capillary walls in the blood, in the brain capillaries, which are tightly joined endothelial cells and don't allow molecules which are charged or not highly lipophilic to penetrate the brain. So the idea we developed is to combine the drug with the targetor, which is the important function, which has two different forms. One is lipophilic, the other is highly water soluble. And here it is the difference, a permanent positive charge placed on the target. So the lipophilic combination, the CDS, is allowed to easily pass the blood-brain barrier. And in the whole body, this conversion to the positively charged occurs. The ones outside the brain eliminated fast, which is highly water soluble. In the brain, the positively charged species is locked in. Now it cannot come out. And then undergoes a various enzymatic conversion to the active drugs. The target of which we are using is a dihydropyridine, pyridinium quaternary salt system. Here it is trigonelline, which is exactly analogous to the NADH, NAD plus coenzyme system in the body. One example given with estradiol, which is important for the well-being, particularly on female subjects. Estradiol has to be a certain concentration to allow normal function of the body. And postmenopausal women have very significant reduction in estradiol concentration. What people normally do now, administer estradiol to replace, but this is not the way to go, because in order to maintain brain active estradiol levels, you need high concentrations in the blood, which can lead to various side effects. So we coupled estradiol with a dihydrotrigonelline, which is then penetrating the brain. You can see the log P that is representing the lipophilicity, 4.5. The oxidation conversion to the positively charged species dramatically changes that by 5 orders of magnitude, which is a truly dramatic change, and that locks in the brain. At the same time, elimination in the body. And then this compound and enzymatic process yields the active estradiol in the brain and some in the blood. Now, this composite is subject to some stability issues, and we stabilized it in form of a complex, 1,2 complex with hydroxypropyl beta-sarcodextrin. And this is actually the drug the FDA allowed us to study and develop. To illustrate the difference produced by this transformation, you can see the half-life of elimination from various organs, all pretty fast, except the brain, which is dramatically different and keeps it in. This is the target or positively charged form. Now, the release of the estradiol was demonstrated by determining immunoreactive estradiol in the brain. Various components of the brain are the same, so it's not specific to certain areas. And at the same time, of course, in the blood, it's eliminated very fast. Taking this drug then development, and here is a study in postmenopausal women where produced highly potent activity, restored the normal function of the brain in terms of cognitive function and hot flashes, but in the same time, the blood levels are maintained the normal level. Same system is applied to peptide brain targeting, where the peptide is packaged between a lipophilic functional group the targetor, but here we use the spacer also to allow release of the peptide. So the spacer is some specific combination of amino acids. And the system works the same way as before. And there's a diol delivery to the brain, lock-in, then various enzymatic processes yields the active target, which is Dudley, D-alanine, D-leucine, and enkephalin, which is an analgesic. And here you can see that the Dudley itself at this concentration don't show any analgesia, but combined as a chemical delivery system is very active. To demonstrate that this is clearly brain targeting, the activity can be antagonized by naloxone, as shown here, and naloxone ion doesn't have any effect. So naloxonium ion doesn't go to the brain. So this process actually was included the top 10 discoveries in '92 by the Harvard Health Letter. Let's see now the other side of the retrometabolic drug design loop, the subdrugs. Here the idea is to design an analog of the drug, which is isosteric isoelectronic, which does not go to the various metabolites like a drug, but will undergo one-step deactivation to a known inactive metabolite, which is converted into the subdrug by chemical transformation, which are designed in a way to produce an active analog and to undergo easy metabolism. Now, this is a process of drug design and development which easily can be converted to computer programs where we take a lead structure and based on transformation rules, the computer will produce a library of inactive metabolites and also active metabolites. But we take the inactive metabolites and other transformation rules, produce a library of subdrugs. And then we calculate the properties of these and rank them and also predict the rate of hydrolysis. And this is how it's easy to identify the best candidates for the subdrug. The ranking factors include molecular size, electronic properties, solubility properties, and all these enter into an equation which describes the relative ranking factor. And these are actually based on molecular orbital calculations. We develop algorithms to predict the ranking of all these analogs. So let's see one concrete example in the glucocorticoid therapy. Glucocorticoids are well known. Multiple drugs have many uses. Glucocorticoid receptor, however, is ubiquitous, is everywhere in the body. And in this way, it's very difficult to target specific organs. For example, in the eye, glucocorticoids produce two major side effects, glaucoma and cataract. So in order to avoid it, we have to design a drug which does not produces the system what is responsible for this toxicity and also to avoid systemic toxicity. Now here is hydrocortisone or cholesterol, which is a normal natural steroid. You can see the multiple metabolism this undergoes, at least 15 metabolites. Now in development, we use proteanic acid in which one of the pharmacophore is destroyed and the dihydroxyacetone side chain is converted to an acid. This quaternic acid has virtually no corticosteroid activity, only 1/10,000th of cortisol. Now what we do, we modify the quaternic acid structure to reproduce at least partially the pharmacophores in a way to have an active subdrug. However, this should then go back to the inactive metabolite, the quaternic acid derivative. For development, we chose the analog of prednisolone. Here is the inactive metabolite, the prednianic acid, which is then converted to a subdrug called loteprednol etabonate. Here we do the two major changes which are novel for this kind of molecules. Introduced the 17-alpha carbonate and the chlorimethyl ester. These together produce the active pharmacophore. But in one step, it will be deactivated to the quaternic acid derivative. Without going in all the details, we can see that loteprednol etabonate and very active glucocorticoid clobetasol propionate have virtually the same pharmacophore structure. This is the chlorine from both overlapping. The side chain 17-alpha position are the same. And if you compare activity and toxicity of various known used potent corticosteroids, you can see that the more potent, the more toxic. So the ratio, the therapeutic index is always about the same. Loteprednol etabonate has a therapeutic index of 24. So it is potent, but much more safe to use. And the loteprednol etabonate became a drug on the market in 1998 by Bosch and Lomb. Bosch and Lomb developed a number of various formulations of these drugs during the patent life. But after the patent was expired, a number of other companies got in the market. And the reason I'm showing it that it started in 98, and you can see that even this year, in January 25, a new formulation was introduced by another company. More than this, you can see that in India, for example, there are actually 37 different pharmaceutical formulations of loteprednol etabonate. So it's worldwide, it's a highly used, very safe, very potent corticosteroid. Now let's see one other example. Again, a receptor which is ubiquitous found everywhere in the body. This is a muscarinic receptors, which mediates the action of acetylcholine together with nicotinic receptors. There are 5 molecular subtypes of these receptors responsible for various functions in the body. The problem is that there are no selective antagonists or agonists known. So whenever you are trying to treat certain muscarinic receptor, you have all kinds of side effects, including arrhythmia, dry mouth, photophobia, etc. And for this reason, the local anti-secretory activity, that is like excessive sweating, hyperhidrosis, is not treated, cannot be treated because of the usual side effects. In order to prevent this, we have designed soft analogs of anticholinergics. Here we show one of them, glycopyrrolate. And these and others have all the same kind of pharmacophore. Is a sterically hindered acid, carboxylic acid, and the quaternary ammonium alcohol. We have tried to develop various subdrugs modifying the structure, introducing hydrolytically sensitive functions, or soft analogs shortening the distance. And all these showed pretty good activity, but for one reason or another, they were not satisfying all the requirement. So finally, we succeeded by modifying the quaternary nitrogen function in the molecule, and it is glycopyrrolate has an activity of 8.6. The corresponding soft analog has similar activity, but it has a better selectivity M3, M2, which is desired. And the hydrolytic product, the zwitterion, is more than 12 times less potent. So this satisfied the subdrug design process. The compound which was developed is called now sofpironium bromide, and here I easily illustrate the activity. Here are sweat glands in the dermis, which are activated by acetylcholine to produce sweat. Now, sofpironium bromide binds to the receptor, successfully compete with acetylcholine, preventing the sweat gland to release the sweat. But in the same time, whatever is getting in the general circulatory system is hydrolyzed fast, so there are no peripheral side effects of the drug. As we have found actually in human plasma, it has a half-life of 11 minutes, and is specifically hydrolyzed by paraoxonase 1, which is an enzyme hooked to HDL. It's not tackled by the expected butyrylcholinesterase, so this is a very successful and lucky actually design. The hydrolysis quantitatively yields the zwitterion, which is eliminated fast. So this molecule went through the usual development of many years, and finally last year it was approved by the FDA here, named Sovdra, in June 2024. The first prescription was issued in December 24, and now it's on the market here. Previously was approved in Japan, is on the market as E-Clock, and next month it starts marketing in South Korea as well. Just to mention, this general concept of the subdrug design is used now everywhere in the world and various fields, from beta blockers to analgetics, anesthetics, benzodiazepines, etc. Just to demonstrate that how everybody is using this. This is a very recent publication by Novartis to the end of 2024 called Oral Subdrug Strategy to Engineer GI Stability and Limit Systemic Exposure to Treat Cryptosporidiosis, which is a diarrheal disease. Finally, one more molecule I wanted to show, which is not a subdrug, but uses the type of protection which we have done for the estradiol delivery system. That is a cladribine hydroxypropyl beta cyclodextrin complex. Called complex because a mixture of two complexes, inclusion and hydrogen bonded complexes. We have developed this many years ago. Some of the initial animal and human studies were done actually at the Pharmaceutical Research Institute in Hungary and Budapest. The molecule is called Mavenclad. It is the first oral multiple sclerosis treatment to provide 2 years of proven efficacy with a maximum of 20 days of treatment. It was approved in 80 different countries, exceeded the equivalent of 1 billion cells per year, so it is called a blockbuster. Phase 4 studies demonstrate the consistent safety and high efficacy. By now, over 100,000 patients were globally treated, and the results from phase 4 study evaluating patients on Mavaclan with highly active recurrence confirmed that 79.2% achieved no evidence of disease activity during year 4 of the treatment. These studies are ongoing. We don't even know how long the efficacy lasts, but want to point out that the drug is treated, or patients are treated 2 times 10 days, 1 year and the second year, and that's it. In conclusion, wanted to show way how to design new drugs to improve the activity by including the activity toxicity ratio as the target. Incorporate metabolic, toxicological, and targeting consideration from the beginning in the drug design process. This is done by combining structure-activity relationship with structure-metabolism relationships. The retrometabolic design approaches have 2 major classes: soft drugs and chemical delivery or chemical targeting systems, and special computer programs are available to help the design and ranking of novel drug candidates. I have quickly showed 3 drugs, new drugs, which developed in our laboratories, which reached the worldwide market. Thank you for your attention.